Dear students, welcome to 11th class Biobotany. So we were discussing about the first chapter Living World. Last class we already covered bacterial structure, bacterial characteristic features and also shapes and flagellation in bacteria. Today we are going to study about ultrastructure of bacteria. So you can see here bacterial cell and what are the components of bacterial cell, and we will discuss in very detail. So you can see here blue color, which it is a kind of a layer that is called capsule, then cell wall, plasma membrane, and plasma membrane has enfoldings which is called mesosomes, and inside the bacterial cytoplasm you can see DNA, circular DNA present, plasmid present, polyribosomes present. We will discuss one by one in very detail. So first of all we are going to study about capsule. So this structure, this is marked as capsule or glycocalyx. So you can see here around the bacterial cell capsule or glycocalyx is present. So what it is actually? Capsule or glycocalyx, which is a gelatinous substance which is made up of polysaccharide or polypeptide or both, which give protection from desiccation and from antibiotics. Okay, and if the glycocalyx, which is tightly attached with the bacterial cell wall, which form the capsule. Okay, and the capsule which helps to retain the nutrients in the bacterial cell, and also because of its sticky nature, it also helps the bacteria to attach on the plant root surface and also human teeth surface. So this is capsule or glycocalyx. So what it is actually? It is a gelatinous substance and it is made up of polysaccharide or polypeptide or both. And what is the function? Which give this glycocalyx or capsule, which give protection from desiccation and also from antibiotics. You already know what is antibiotics. These chemicals which kill bacteria. So the capsule covering which protect the bacteria from actions of antibiotics. Okay. So that's about capsule. So you can see here outer layer, that is capsule or glyco. The next component of bacterial cell, which is bacterial cell wall. Bacterial cell wall is granular and rigid, which give protection and shape to the bacterial cell. The chemical composition of bacterial cell wall, which is made up of peptidoglycans. So what it is actually? Peptidoglycans, which means which contain polysaccharide parts and also peptide part. Okay, glycan which is related to saccharides, polysaccharides, and peptide which is related to peptides. So it is actually peptidoglycan, or it is also called mucopeptide. So this mucopeptide, which means the structure or chemical composition means which consist of N-acetyl glucosamine and N-acetyl uramic acids. They are connected and they form layers N-acetyl glucosamine, then next N-acetyl uramic acid, and one layer of N-acetyl glucosamine and N-acetyl uramic acid, which are connected to the next layer of N-acetyl glucosamine N-acetyl uramic acid by a peptide bond. Okay, they are cross linked by peptide bonds. So this complete structure which is called mucopeptide. So this is the chemical composition of bacterial cell wall. And this bacterial cell wall, a polypeptide is present, that is pore rings are present, and these pore rings which helps in diffusion of solutes. So these are the bacterial cell wall. You can see here, this is bacterial cell wall. So suppose what it means mucopeptide, once again mucopeptide or peptidoglycan, which is the chemical composition of the bacterial cell wall. So suppose this is an acetyl glucosamine. I wrote here like NAG. Then next one N-acetyl uramic acid. Then again N-acetyl glucosamine, N-acetyl uramic acid. The next layer N-acetyl glucosamine, N-acetyl uramic acid, then again N-acetyl glucosamine, then N-acetyl uramic acid. And this layer and this layer, they are cross linked by peptides. Okay, the peptides of peptides which is made up of 4 to 5 amino acids. Okay, and this all structure it is called mucopeptide or peptidoglycan. So this all structure which form the bacterial silver, that is made up of mucopeptide or peptidoglycan. Bacterial plasma membrane. Bacterial plasma membrane which is made up of lipoprotein. So you can see here, this is the plasma membrane of the bacteria made up of lipoprotein. So animal cell and plant cell plasma membrane also made up of lipoprotein, same in the case of bacteria. And the plasma membrane which control entry and exit of small molecules and ions. Usually plasma membrane is semi-permeable, which does not allow the every molecules. They control the entry and exit of small molecules and ions. And the plasma membrane, the enzymes of on the plasma membrane or in the plasma membrane, the enzymes which involved in oxidation of metabolites, like that. For example, enzymes which involved in respiratory chain, and also photosystems of photosynthesis also present on this plasma membrane of bacteria. Bacteria, they do not have the membrane bound cell organelles mitochondria and also chloroplast. Okay, so the enzymes which is responsible for respiratory chain or cellular respiration, which is present on the plasma membrane. Actually, in our cell, animal cell and plant cell, which is done by the help of mitochondria. But bacteria, they do not have mitochondria, so all these enzymes are present on the plasma membrane. Same, the photosystems of photosynthesis, they are also present on the plasma membrane, because bacteria, they do not have chloroplast. So, and the photosystems of photosynthesis also present on the plasma membrane of the bacteria. So these are the main points about plasma membrane. Bacterial cytoplasm. So bacterial cytoplasm, you can see here, this is bacterial cytoplasm. In this, which is thick and semi-transparent. And in this cytoplasm, ribosomes are present, which is 70S ribosome. Okay. And also cell inclusions also present in this bacterial cytoplasm. For example, glycogen, poly-beta hydroxybutyrate and sulfur granules, and also gas vesicles also present in this bacterial cytoplasm. So bacterial cytoplasm, which is thick and semi-transparent, and in this ribosomes are present, and also cell inclusions also present. Bacterial chromosome. We already discussed bacteria, they are prokaryotic organism. Why they are called prokaryotic organism? Because they lack nuclear membrane and also membrane bound nucleus and also membrane bound cell organelles. So bacterial chromosome, which is a single circular DNA which is tightly coiled without any membrane. Okay, so you can see here bacterial DNA here. This bacterial DNA or this genetic material is called nucleoid or genophore. Okay, and it contains all informations about this bacteria. And this bacterial DNA, which is not bounded by histone proteins. So in normal eukaryotic organism, histone proteins are present, which on which DNA fragments are wrapped. Okay, so such kind of these histone proteins are not present in bacterial cell. Okay, so it is simply a circular DNA, and which is highly coiled and which is not bound to histone protein, and which contain the informations about the bacteria, and which is sometimes which attached on the plasma membrane. The one end of this bacterial DNA which attaches on the plasma membrane, and which is make easy to separate the DNA fragments at the time of cell division. Okay, so bacterial DNA, these are the main points. Bacterial DNA or genetic material is called nucleoid or genophore, and there is no membrane around this DNA or genetic material. So bacterial chromosome, they are simply stand in this cytoplasm. Plasmids. What is plasmid? Plasmid is extrachromosomal DNA which is double-stranded and self-replicating and autonomous elements. So this is plasmid here. You can see in the bacterial cytoplasm, which is the extrachromosomal DNA, and it is self-replicating and autonomous. That means they can make their own copies without any help from bacterial chromosome. So they make their own copies and they contain the genes responsible for fertility, antibiotic resistance and heavy metals resistant to antibiotics and heavy metals. Okay, so they contain the genes for fertility and genes for resistant to antibiotics and heavy metals. And the number of plasmids in the bacterial cell also varies, and the size of plasmid also varies. It may be 1 to 500 kb. Okay, and the based on function, the plasmids present in the bacteria are classified into different types. What are those? First of all, we have discussed the major function, fertility. So the plasmids, the number one, F factor or fertility factor. It is a type of plasmid. The second plasmid, resistant plasmid, or it is also called R plasmid. R plasmid, which means resistance plasmid. And the third one, cold plasmid or coalescent plasmids. Okay. And fourth one, Ri plasmid or root inducing plasmid or Ri plasmid. Then the last one, Ti plasmid or tumor inducing plasmid. So this plasmid, they have the genes for responsible for fertility and antibiotic resistant heavy metals, and also they produce bacterial toxins and bacteriocins, etc. Okay, so they are extra chromosomal DNA and they are double stranded and they are self-replicating autonomous elements. They can make their own copies and their number varies in bacterial cells. Bacterial mesosomes. So what is actually mesosomes? Mesosomes are the enfoldings of plasma membrane. So you can see here plasma membrane and this structure, the enfoldings of plasma membrane, which is called mesosomes. So mesosomes which present in the plasma in the bacterial cell in the form of vesicles or maybe lamellar or maybe tubular structures. And what is the purpose of mesosomes? You know, bacteria, they are very small, they are unicellular. Okay, so they have to perform respiration for getting energy. Okay, and they also want to do binary fission. So they have to increase the surface area for these functions. They have to increase surface, they have to maximize the surface area for respiration. So how this possible? Only possible by enfoldings of plasma membrane. So they increase the surface area by forming mesosomes in the bacterial cell. That is called mesosomes. Okay, so mesosome, they maximize the surface area for respiration and also for binary fission. So this is mesosome in the bacterial cell. Polyribosomes. So ribosome, you already know, ribosomes which help in protein synthesis. Okay, they are site of protein synthesis. So ribosome, what type of ribosome present in bacteria? Basically there are two types of ribosome, 70S ribosome and 80S ribosome. 80S ribosome present in animal cell and plant cell, and 70S ribosome present in bacteria. Okay, 70S. So this is the structure of a ribosome. They have a small subunit and large subunit. Small subunit is made up of 30S and large subunit it is 50S. They together form 70S complete structure, that is 70S ribosome. So they are the site of protein synthesis, and the number of ribosome in bacterial cell also varies. Maybe ranges from 10,000 to 15,000. Okay, if many ribosomes, if they are held together by mRNA, this complete structure is called polyribosome. So what it means, polyribosome? Poly means many ribosomes. So many ribosomes are held together with the mRNA, that form polyribosomes. Okay, so ribosome, what type of ribosome present in bacteria? 70S. What are the subunits of this ribosome? 50S and 30S. Bacterial flagella. So you can see here bacterial flagella here. So what it is? Bacterial flagella, they arise from bacterial cell wall, and this is like a thin hair-like structure which is present in motile bacteria. So this structure which helps in locomotion. Okay, and this bacterial flagella which is made up of a single fibril and which is length its length is around 20 to 30, sorry, 50 micrometer, and diameter is around 20 to 30 micrometer. But eukaryotic cell also, they have also this type of flagella for locomotion, but that is different from bacterial flagella. Eukaryotic cells flagella, it is made up of 9 plus 2 microtubules, but bacterial flagella, which is made up of a single fibril and which emerge from the bacterial cell. Okay, so this is bacterial flagella, which is present only in motile bacteria, and flagella helps in locomotion. Fimbriae or pili. So what it is? Same like our flagella, these are hair-like appendages present on the surface of cell wall of the gram-negative bacteria. Okay, they are present on the gram-negative bacteria cell wall. For example, in a gram-negative bacteria, enterobacterium. So in this type of bacteria on their cell wall, hair-like appendages are present there. That's called fimbriae. And this length is around 0.2 to 20 micrometer, and diameter is about 0.025 micrometer. And some bacteria, they have a special type of fimbriae present, that is called sex pili, or sex pili, which helps in conjugation. Conjugation means the transfer of genetic material from one bacterium to another bacterium, which is done with the help of sex pili. Okay, so this is about the structure sex pili. So these are present on the surface of cell wall of the gram-negative bacteria.